All right, it's 1 minute after the hour, so why don't we get started here? First, I want to welcome everyone. Thanks for taking time on a Thursday afternoon to hear a little bit about rock snot and Didymo and the issues that we're having it up on Lake Superior and in our North Shore streams. I want to welcome everyone, especially the researchers that are involved in these projects, as well as all of our partners. This includes our agency, tribal, nonprofit foundations that are joining us today. And really, I got an email about a month ago from someone that said was addressed to Didymo enthusiasts. So I think that's actually who's joining us today, are all the Didymo enthusiasts out there who have some knowledge of Didymo and some of the resource issues that it poses for us. Today's plan, we're going to go for, hopefully we can wrap, we're planning to wrap this up in an hour. We're going to have sort of two parts of today's plan. We're going to start with a welcome and introductions, history of the problem, talk about our research strategy on the North Shore, and then talk about what we learned in 2021 and what we're planning to do in 2022. After that, we're going to open up for questions and results. Ask questions about Didymo, ask questions about some of the results that we talk about, and ask questions about the plans. Heidi Randala from DNR is going to be moderating that Q&A session. After we do that for, we'll see, 10 minutes, 15 minutes, whatever we feel like, we're going to switch to education and outreach. What we want to do is have a discussion about how we can help each other get the message out. We'll talk about things that we've done in 2021 and then look forward into the future, how we can all get together in 2022 and beyond to get that message out there. We'll finish that up with a Q&A and a discussion period to talk about the needs and opportunities that exist for Didymo. Well, let me start by introducing our research team. I think most of them are on the line today, so you can match their faces to their pictures here. But from the Science Museum of Minnesota and the St. Croix Watershed Research Station is myself, Mark Edlund, Adam Heathcote, David Burge, and Farzad Sarjadi. Farzad is a member of the STEM Ed group and also looking forward to some of the outreach that we're going to be doing on the project. Representing Minnesota DNR Fisheries is Heidi Rantala and Corey Goldsworthy. Heidi is a amazing stream biologist. Corey is Lake Superior Fisheries, and between the two of them, we can find that intersection between what happens between the streams and the big lake. Bob Pillsbury joins us from the University of Wisconsin, Oshkosh. Bob has experience working on Didymo in Lake Superior, and especially in eastern Lake Superior, where the St. Mary's River has some problems. He's helping us out with molecular analysis of bacterial communities associated with Didymo mats. At the top right is Cody Sheik, professor at the University of Minnesota, Duluth. Cody is welcoming a new grad student in the fall, Sarah Klaus. Sarah is going to be a student in the WRS program and will be also joining us on our Didymo efforts, primarily possibly looking at nutrient cycling and 18S molecular work on Didymo mats. And finally, less than 2 weeks ago, Joe Mohan joined us as a postdoc on the project. Joe is a postdoc at the Science Museum of Minnesota out at the St. Croix Watershed Research Station with us, and he is cutting his teeth. I think I had him counting Didymos on his second day in the office. So I expect most people have heard of Didymo if they're on the phone call, of course. And but if you haven't, Didymo is a microscopic alga, but it's a big microscopic alga. It's a big diatom. It tends to be about a tenth of a millimeter long, still microscopic, but for diatoms, that's a beast. Where it normally lives in the world are in cold water, low nutrient streams and lakes, often at base flow conditions. We know we find it in Lake Superior, as we'll learn. I've seen it in Lake Baikal, Lake Hovsgol in Mongolia, as well as streams in Alaska and things like that. However, in recent decades, it has been appearing around the US, around North America, and around the world. in places where it's not been noted before and growing in ways that are disconcerting. What happens with Didymo is it can form these very, very thick mats of mucilage. The growth of Didymo is based on what you see on the right side of your screen there. Each Didymo cell is living at the end of a large mucilage stalk. And so most of what our New Zealand colleague there is holding is mucilage stalks with a thin layer of Didymos growing on the outside of them. Why these things are getting around the world, why these things are being introduced in various places in the US is typically, we're looking at two hypotheses. One, is there an aggressive colonization that's occurring? So new strains of Didymo that are being introduced and into these habitats? Or are our environments just changing to become hospitable to Didymo and that the Didymo can thrive in these environments? Where the Didymo gets introduced and becomes problematic, what it does is it represents sort of a triple threat. It affects the ecology, it affects recreational opportunities, and it affects economies. From an ecological standpoint, we know that where Didymo is introduced, it changes things. It changes the bacterial communities associated with the algal mats. It changes the actual algae that are living there. It changes the invertebrate communities. And of course, this all gets transferred up to changing the fisheries communities in these streams. From a recreational standpoint, it's a bit unaesthetic. It looks like brown goo coating every available habitat in streams. And it creates problems for anglers with ability to changing both the fisheries and ability to cast and retrieve their lines. And it kind of ruins the opportunity for others who are trying to enjoy some of our resources. Last is the economy. Because of these first two things, it affects the economy actually. In New Zealand, they talk about multi-million-dollar losses to the tourist economy because of the Didymo introduction there. We've got a $250 million North Shore summer economy that is potentially at risk because of these changes in Didymo. Missed one slide there. So let's start with what we know about Lake Superior Didymo. I'm going to start with a wedding picture because that's where in Lake Superior it actually all starts. That's a wedding of my PhD advisor, Gene Stormer, on his honeymoon in 1961. They did the circle tour of Lake Superior and stopped in Grand Marais, and he took a diatom collection on Artist Point. As far as we know, that's the first report of Didymo from Lake Superior. He published from that collection a couple different papers, and we also know that Didymo was also found in Lake Michigan in the 1800s. Again, suggesting us not that Didymo appeared in 1961, it's just that no one looked for it before 1961 in Lake Superior, and I would suspect that it was there. From the 1960s to the 1990s, we knew Didymo was out there. It was part of a number of periphyton studies that took place up out of Duluth. And even on the Keweenaw Peninsula, Mary Moffat did a beautiful study on transmission electron microscopy of populations from that part of Lake Superior. So we knew it was there, and sometimes it was reported as Gomphanema geminatum instead of Didymosphenia geminatum. Then things changed. In the late 2000s up through the 20s, we started noting increased abundances of nearshore Didymo, to the point where we saw headlines like, "That's not toilet paper, it's rock snot." Those efforts, or those acknowledging that we're seeing some changes in those nearshore communities, initiated Joe Thompson, Doug Jensen from EPA and UMD, put together a sampling effort and a white paper documenting the distribution of Didymo up and down the North Shore, as well as the abundances of it. And it began at that time then some opportunistic survey efforts by a bunch of us, started keeping an eye on what we were seeing out there, but recognizing that periodically it was showing up as a bit of a problem in nearshore Lake Superior. And then 2018 happened. In late summer of 2018, some folks noticed some odd growths in the Poplar River. And managed to get some samples out to take a look at, and it was the first ever confirmed report of rock snot, which is what the media calls it, or Didymo, in a North Shore stream. It got a wide press because of the effect that it's had on other river systems throughout the U.S. And it really was spurred on our interest in further work on it. When we put that stuff under the microscope, it was clear these Didymo were not a lake, a Lake Superior mat that someone had carried up and thrown in the stream or something like that. These guys were happy and living there as a happy, healthy Didymo community. Again, that between the Poplar River invasion and the changes in Lake Superior really spurred our group's efforts to work on this Lake Superior and North Shore Didymo threat. We wanted to address these two hypotheses. Is it aggressive colonization of a new strain that's getting around, or is our environment just becoming conducive to Didymo growing in these places? Together, we reached out to a number of different funding agencies, and definitely want to acknowledge the contributions that they've made to our efforts. LCCMR and the Environment and Natural Resources Trust Fund, the Minnesota DNR Coastal Program, and the Minnesota Sea Grant have each funded different aspects of the project that I want to talk about, and again thank them for their contribution to our efforts. So what are these projects that we're going to work on do? First, we want to determine what the source of Didymo in these north streams in Lake Superior are by comparing those samples to one another, Lake Superior and the North Shore stream samples we run up with, and to North American population using molecular techniques. Secondly, we want to understand the distribution, seasonal dynamics, and the effect of Didymo on these habitats. This is going to be done through some monitoring of 5 sort of premier stream and lake sites from 2021 to 2023. And these sites are going to be hit for water quality, algae, bacterial DNA, and ecological condition. Third, we want to understand what other North Shore streams may be at risk. So in each of those years in 21 to 23, we're going to be sampling another 20 of those streams and lakeshore sites, doing a full measure of what we were also measuring in the other 5 more detailed survey streams. And finally, with our DNR coastal survey, we've actually moved our efforts out into Lake Superior for the first time. We're going to be looking at monitoring transects, depth transects along known spawning reefs out in Lake Superior to look at distribution of periphyton and Didymo out there, as well as its potential impact on the fisheries that use these sites, including coaster brook trout, lake trout, and our coregonid whitefish and cisco fisheries. There. So what did we do in 2021? Our funding came in August of 21 and August of 2021, and by the tail end of August, we were up sampling our streams. We started out with a 5-stream nearshore survey. The stream sites that we're targeting for the next 3 years will be the Lester, the Knife, the Caribou, the Poplar, and the Devil Track. At each of these sites, we measure physical characteristics. We measure temperature, pH, DO, chlorophyll, all the things that we measure at in stream and nearshore sites. We're collecting periphyton using these beautiful lobe samplers that were designed and built by Chris at the DNR French River. We use those to look at ash-free dry mass, benthic chlorophyll, diatom communities, and of course molecular samples to look at sources of Didymo. And in each of these 5 streams, we've also deployed loggers to get full or temperature and discharge records year-round on these sites. Come on. Wow. In the other thing we're doing is in 2021 was our Blitz survey, where we hit 20 additional streams and measured the same types of characteristics of each of those streams, just not seasonally. And those reached it from the Lester River all the way up to Grand Portage Creek. Finally, getting out into the lake, our 5 fisheries transects were sampled in September and October of 2021, included Fitker's Reef, reef off of Stony Point, reef off of Gooseberry, and Taconite Harbor, and Grand Marais. These were transects that extended from 2 meters depth out to 16-meter depths. In each of these sites, we located great reef habitat, collected water quality, ash-free dry mass, paraphyton samples for molecular analysis, and using a wonderful dive team and the RV Blackfin. And then in October, we also deployed egg mats to try to assess our spawning in these sites. So what have we learned in 2021? Probably the most shocking thing that we learned is that Didymo was found in 7 different streams along the North Shore. These range from the Carlson all the way down to the Caribou. And we found varying abundances of these. Places like the Flute Reed, we found just a very few cells. Other places we found up to 100,000 cells per centimeter squared. These are high abundances of Didymo. One thing that's interesting is the abundances were much, much greater than the abundances in the nearshore area of Lake Superior at several of the river sites. Interestingly, we did not find Didymo in the Poplar River in 2021. Well, we did find it, of course, very commonly along many of the sites that are listed on the right there, along Lake Superior as well. What does Didymo look like? Again, I got 3 pictures here from Didymoan rivers. They include the Caribou River, the Devil Track, a couple pictures from the Devil Track. But you can see in the Caribou, these little tufts of fuzz on the tops of the rocks are sort of early Didymo mats, whereas in the Devil Track River, we can see that these rocks are completely coated with Didymosphenia mats. On the right, there were many streams that didn't have Didymo, and we expected to see algal or paraphyton communities look like you see on the right there that did not have Didymo. Other things that we learned. 2021, which is when we found it in 7 streams, and 2018 when it was found in the Poplar River, are some odd water years. Both of those years, in comparison to other typical years, had very, very long base flow conditions throughout most of the summer, and also were prefaced by either a very rapid spring freshet in 2021, coming right back down to base flow, or in 2018, actually, most of the snowpack was lost in October before the water year started. So the low base flow conditions are obviously well known for as part of our encouraging Didymo to grow, and also seems to be a case here on our North Shore stream. We're still analyzing all of our water quality samples. We've got some data in, but generally along our North Shore streams, we're seeing low nutrient conditions of both soluble reactive phosphorus as well as our nitrite, which is typical of North Shore streams, and again, something that we might expect when we see Didymo appearing in places. Other things that are ongoing, Bob Pillsbury at University of UW Oshkosh is handling the 16S bacterial analysis. Those are underway right now for our 2021 samples. Bob's done previous work on the St. Mary's River. The graphic on the right shows just how the bacterial communities differ between paraphyton or algal mats that have Didymo and paraphyton mats that don't have Didymo. And I would think we may see something similar in our North Shore streams as well. Other molecular work that's ongoing is on the Didymo and its sources as well. David Burge and Cody Sheik are heading up that effort. using two different approaches. First, and this is in partnership with the University of Minnesota Genomics Center. First, they're doing long-read DNA sequencing of two populations, one from the lake and one from Kimball Creek, actually, where we've individually isolated 50 Didymo cells, and we're going to be doing long-read DNA sequencing of those to first identify what genetic differences may exist between these lake and river populations, and then to also create a reference genome of Didymo, so with which we can compare other populations from Minnesota and North America too. The second strategy with molecular for Didymo is going to be directed at our other collections of Didymo from Minnesota, as well as from throughout North America. What we want to do is use shotgun sequencing here and compare those sequences to our reference genome from our long read work to identify intraspecific and strain biogeography and phylogeography to hopefully identify what the relatedness of our North Shore and Lake Superior Didymo are to other populations from throughout North America. On our fisheries transect work, this is stuff that had never been done before. So first we were able to develop methods for sampling that were diver deployed, working with a great dive team. What we found out in Lake Superior was in most of the transects, very nice heavy periphyton growth out to about their 16-meter depth stations. However, although there was good growth of algae out there, the Didymo itself was limited to the very near shore stations in our September and October sampling. We did have some success collecting eggs. Those are being analyzed by Lauren Miller with the DNR. And we did learn from this experience that we needed to modify a bit of our approach to this, which I'll talk about for our plans for 2022. So last, what are our plans for 2022? In May through October, we're going to initiate our monthly 5-stream survey that we talked about from last year. We're going to do the same rivers: Leicester Knife, Caribou, Poplar, and Devil Track. In July, we're going to couple our July sampling with our 20+ stream or near shore blitz. We're moving it a little bit earlier in July than we did last year in August. We had actually a sense that in several of the streams, the Didymo mats were actually sloughing off already in August. So we're thinking we're going to hit that a little early, and really appreciate Margaret Watkins up at Grand Portage Band has partnered with us to hopefully get us onto some of the tribal streams in July for that sampling, which we weren't able to do last year. In July through October, we're going to be doing our fisheries transect sampling out on Lake Superior. We're dropping one out of the mix because of some logistical constraints with finding weather days and diver fatigue. But we're going to be doing our Fitkers transect, our Stony Point transect, our Gooseberry River transect, and our Grand Marais transect. To enhance our ability to, or better our ability to understand spawning, we are going to deploy larval fish light traps to look at spawning success in both May 22 and May 23. And in the fall, rather than the egg mats, which couldn't tolerate some of the physical conditions out in Lake Superior, we're going to use a vacuum technique to collect eggs. And these, again, will be coupled with some fall gill netting surveys along these sites as well. And finally, in October, we have a new DNR coastal funding for some additional stream work that has been recommended for funding. This is going to be based on doing some longitudinal surveys up and down the streams. The Cross, the Brule, the Devil, and the Kimball Creek, and all of these are known coastal brook trout streams. We're also adding some invertebrate work on this sampling. And one of the things that we have learned from that first year is that Didymo is not everywhere, and so this is our first chance to get a better sense of what the longitudinal survey, or what the distribution is in longitudinal in these stream sites. So that is what we've it gives us sort of the basics of what we know about Didymo and the world in Lake Superior. its appearance in the streams, what we're tackling with our research ideas, what we accomplished in 2021, and what we accomplished in what we're hoping to accomplish in 2022 with our field season. What I'd like to do is open it up for some Q&A. Heidi'll moderate things. You can use the chat feature in Zoom down at the bottom. You click on that and type a question in if you'd like, or feel free to go into the reactions thing, and you can raise your hand down at the bottom as well. Use that feature. So if people have questions, please, we'd love to discuss what we've found so far. We thought I was concerned when we went out in 2021 that we were going to find nothing in the streams, and I tell you, I think we were all shocked at what we did find. That Didymo was prevalent in a number of streams and in very high abundance in those streams. So
it looks like we have a couple questions in the chat. Going in order, Elena said that is wondering if Cook County SWCD can assist with sampling. And she said that there's volunteers that would probably like to help.
That would be awesome. A little later, I'm going to show you what our sampling schedule is, and please sign on the dotted line.
And Travis from Grand Portage State Park asks if we're planning on including the Pigeon River in the sampling.
Travis, you're on my list of contacts, and we are going to get up there for that fall or that July sampling, and would love to connect and sample the pigeon as part of our Blitz sampling, if we could.
Okay, yeah, if I can just jump in for a second.
Absolutely.
I was just mainly wondering, because it seems like all the other streams that this stuff is found in, like, I don't know if it's on the Pigeon River. I haven't noticed it myself. But the mouth of the Pigeon River in the bottom, like 1.5 to 2 miles of it, is more of a kind of a muddy bottom and real murky water, as opposed to shallow, clear, and rocky bottom like most of these other streams. So that's mainly why I was wondering if that would have an impact, if it would make it less likely to encounter that on this river, or not. I don't know if that makes a difference or not. Or if it's even,
yeah, I think in general, Travis, that we associate Didymo with hard substrate,
right?
Growing on rocks and logs and stuff like that.
And there's certainly a lot of rock in this river, but it's a little ways upstream to get to it. Yeah.
And I think that would be sort of what we would work to target in sample, and coming up with a sampling strategy with you, is to see if we can access a site that has the right types of substrate there.
Yeah. Okay. Thanks.
And Doug Jensen is on the call, and he's offering a historic perspective with regards to citizens reporting suspicious sightings to him and to Joe Thompson, and their personal observations starting around 2000. And then they looked at different sites from Duluth to Grand Portage to understand the distribution of Didymo. Doug, did you want to add anything to that?
Oh, that was perfect, Heidi. Thank you. Yeah, so that was just to provide kind of a historical perspective on when citizens really started to discover that there was something unusual in the water, and they began to report them to me. And then I also observed them based on some of my own personal fishing activity, and noticed that there was also a change along the shoreline. So by 2008, that's when Joe Thompson and I teamed up to do that reconnaissance to find out if Didymo was part of the issue and what the possible distribution was along the shore, resulting in that white paper. Thanks.
Thanks, Doug. And Liz from Lake County SWCD is asking about the spatial distribution of the mats within the streams. and how far we searched upstream and downstream. And go ahead, Mark.
Oh, you can. Why don't you answer it? Heidi,
Well,
you
I
go
guess
ahead.
I was just going to say that most of our sites were located around Highway 61 for the stream work. And although I had a chance to go out with the Lake Superior fishery crew to electroshock the Devil Track, and gosh, I wish Nick was on the call because he would know exactly the mileage that we saw it upstream, but it was for sure at least half a mile upstream, but it became more patchy. And hopefully the project that David is going to lead will get more at that question. But and also we found it by the mouth in those rivers that we were sampling as well. Other questions? I guess before we move on to the next part, I was hoping, Mark, that you could provide some perspective to effects to other trophic levels from studies around the world, maybe, or other not, or including the US.
Yeah. I think, I mean, in general, the Didymo tends to have an effect on sort of all the levels of the trophic thing. And we showed you some, data from Bob's work on the St. Mary's River, where bacterial communities are, that's about, from a statistical analysis, that's about as night and day as you can get, I think, and how they differ between those two different communities. As we move up the sort of the trophic level, the algae within Didymo mats is a much more homogeneous group, a lower diversity, smaller number of taxa that live within Didymo compared to sort of non-Didymo mats. And there's been several studies that have, sort of quantified, other diatoms that are primarily associated with just, or associated with Didymo mats as well. From the invertebrate work, the tendency is to move away from some of the high profile, high energy, good fish food to more of the chironomid types of benthic inverts. And Heidi, you may have more interest in that. And then last is the fisheries stuff. We know that it affects especially the age structure of fishes when we get Didymo within stream reaches. And I don't know if we know about the spawning. That's one of the things that we're hoping to work on our North Shore studies here. You have anything to add to that, Heidi?
Well, I have a lot to add about the invertebrates, but
Go for it.
Personally, folks, if you want to talk about that, I'm just excited to move up the food web and see what's happening in the animals, especially. I think we're ready to move on to the next part of the presentation, where Mark's going to talk about outreach and education efforts, current and future, and ask for what your needs are. But during that time, feel free to continue to put questions in the chat.
Okay.
Take
All
it
right.
away, Mark.
Okay, thanks, Heidi. Well, we didn't just do field work in 2021. We were also getting the news on the street. We were really taken aback by how much Didymo we found in our surveys of the North Shore streams. It resulted in some initial press, nice work out of Duluth News Tribune, a real nice piece on the from WTIP, and a nice blog post from Quetico Superior Foundation. One of their writers joined us for part of a day of sampling. We've put most of that work into, or most of those initial results into that two-page flyer that I shared with the initial shout out to invite everyone, which you can have, and of course, feel free to distribute that as much. Among the rest of us, we've all been, working to put the news on the street, both also within our own organizations and in some of the some of the other types of social media posts up there. I know I saw stuff on Bob's contributions coming out of the University of Michigan Biological Station, as well as UW Oshkosh. I know we've all talked within our own agencies and organizations about that, and we've begun to reach out to some of the stakeholder groups. Heidi's done some presentations at the Great Lakes Aquarium, as well as with the Arrowhead Fly Fishers group. One of the things that we do want to talk about is the potential for citizen science in this. The DNR supports a program called EdMaps, and this is a place where you can report aquatic invasive species. And we would love for people to, if they're seeing Didymo, to take a picture of it and go ahead and post it on this. Other people, if you haven't heard of EdMaps, you may have heard of iNaturalist and our user of that. iNaturalist communicates with EdMaps, so you can also, if you already have an iNaturalist account, you can feel free to put your Didymo records on there as well, and they will be communicated to us. But we would love to know if people are seeing Didymo out there, where it's occurring, and if we can get it on relatively short notice, feel free to just send an email. It'd be great to get great to get us out there to take a look at it as soon as possible. For 2022, what we really want to do is take our team Didymo and the power of the Science Museum of Minnesota and really become the science and education nexus for all of our stakeholder groups. Put a bunch of images of folks who have shown interest in partnering on our projects, and we want to work together to do that work, whether it's through press, whether it's through communications, whether it's through education. So what we really would like to do is, how can we help each other in our education and outreach activities in the coming years? One thing, just throwing it right out there, we've already plotted out our field schedule for 2022. It's listed right there. That's when our groups are going to be up on the North Shore. If there's any sort of community events that we can work with and collaborate on, that would be awesome opportunities to do that. If someone wanted to go out and do some sampling with us, awesome opportunity to join us in the field and see what this issue is all about. Are there community events? Are there festivals or something like that where a booth or something would be a valuable contribution to that thing? How about messaging? How are we thinking about telling people about this? Is it getting signage on kiosks? Is it trying to talk to groups about this? Is it doing a speaker presentation at one of the state parks if we're going to do that again? Those types of things. We definitely want to encourage the citizen science aspect of it again. Those eyes on the ground or eyes on the waterways or eyes on the lakeshore, whatever, are an amazing way for us to understand what the problems are out there. We are reaching out across North America to colleagues who have access to Didymo collections, because we want to have those for our molecular analyses. If you know people, we've got a list that we've started to put together, but if you know colleagues or friends or whatever that have access to Didymo, what David is putting together are Didymo sampling kits, where we send them out across the country. Don't have to be refrigerated, don't have to be frozen. Basically get the Didymo in there. We're going to preserve it with a molecular fixative and use those to analyze our different populations of Didymo throughout North America. Doug brought up a really interesting question. He was noting that he and Joe Thompson were picking up sort of citizen interest, citizens noting these problems on the shore. If anyone has access to algae collections from historical algae collections. It would be really nice to find out one question that has been posed is, how do you know Didymo wasn't there before? And no one just we just didn't notice it. And I mean, I think we do have lots of folks with experience with Didymo that there's a high likelihood that someone would have seen it. But if we could find some of those collections that might give us some evidence of whether Didymo is just found in low abundance in these streams and just takes off, and funny years like 2021 would be give us a lot towards answering these questions that are that form our hypotheses on there. And then last, it would be nice to we also want to build some educational opportunities around the North Shore and around Didymo. As part of our Sea Grant project, they're supporting at the Science Museum of Minnesota, our STEM ed instructors are going to adapt some existing water science programming to include results from Didymo projects. The STEM ed program at the Science Museum of Minnesota reaches out to all 87 counties in the state of Minnesota with K through 5 programming. And it would be great to, if you have schools, private, public, parochial, tribal schools that would value from this type of engagement, please reach out to us. And we'd love to get up on the North Shore communities and beyond with with our messaging around around Didymo and North Shore ecology. Come on. All right. Well, that was the, I gotta go back one. Here we go. So that that was our sort of our introduction of what we've done for outreach and education so far and what some of our plans are. But really, this is about our our partners and what they can how we can work together. What are the needs of our partner groups? How can we work together, and how can we help each other in these efforts? And I'd love to have a discussion again. Please use the chat or just simply raise your hand in Zoom, and we're happy to hear how we can help each other on this stuff. And Heidi will take care of moderating this as well.
Yep, I just want to note that Doug says that Sea Grant had produced some Didymo watch cards that are available for distribution, and that you can request them by contacting Sea Grant. I know I've requested them via the Sea Grant website. And it looks like Elena has her hand up.
Thank you. I didn't know there's Didymo watch cards, so that's great. But so maybe, I don't know if there's other resource tools, but for our office, because we do a lot with community science outreach and a lot with citizen scientists, especially for monitoring, if there's like kits or packets or anything we can give away, because I do a lot with lake associations, which are just a wealth of people. And we work with some fishermen groups. So if there's any sort of toolkits, for lack of a better word, or anything that we can just have and then we can distribute it, that would be great. And that's just one way we'd be, of the many ways we'd be happy to help out. So I don't know if that's out there, but just let me know.
Liz had a question asking if our research observations were going to be posted to EdMaps. That's a great question, Liz. No, one is, because I tested EdMaps, but I don't think our other observations are. Mark or David, yeah. Or can you respond to that?
Yeah, I can respond to it. I brought our new postdoc Joe in and had him immediately diving in on things. And one of the things that I had him doing on each of these sample collections that we were analyzing that had Didymo in them, I was having him take some images under the microscope with the full intent of getting these observations up on EdMap. I'm also thinking about taking some of our other slides. We did a little bit of scouting ahead of time on these trips, and I'm thinking of actually taking those down going to teach a class on diatoms in May. And I'm thinking of taking those slides down and actually having the students enter the observations as part of this. So those are my ideas. But yes, we will be getting those up on EdMaps.
Elena had posted a question about the education and outreach before she asked the question. Was there anything, Elena, that you wanted to add to that?
Can you send out the dates just for reference?
Absolutely.
Thank you.
And then Brandon says that from Park Services that they have a youth environmental education event in Grand Portage at the National Monument in the week of July 18th. Come on,
Right.
I added some emotion to that. Matt said that Matt from 1854 Treaty Authority said that there is a mention of Didymo mats washing out of streams by August of 2021. Is this kind of a normal life history trait of Didymo, a late summer dieback?
I would say what you're noting, Matt, is probably what we were seeing as well. As I mentioned when I was talking, one of the things that just seemed, it seemed to us when we were out sampling that a lot of these Didymo mats were past. They were sloughing off and things like that. So it doesn't surprise me. I had not heard that people had noted them washing out of streams, but that's a nice thanks for pointing that out, because I had not heard that.
Laura,
Again, that's one reason we're going to move our sort of our Blitz sampling to July so that we can get a full coverage of the whole North Shore system in one fell swoop a little bit earlier than we did last year.
Laura from the AIS program at DNR is asking a question about reporting Didymo to EdMaps and iNaturalist. Is there photo guidelines for taking good pictures of Didymo? What kind of detail do you need in a photo to confirm the identification?
I think the unfortunate difficult thing with Didymo is that it is true. It's although we can see these big slime balls on the stream, we can't actually absolutely confirm it's Didymo without popping it under the microscope. But people have access to a microscope. You don't even, it's a big diatoms. You don't even need a really awesome microscope. You just need a reasonable microscope. And it's actually pretty darn easy to take your cell phone, hold it up to the eyepiece of the microscope and pop a picture of it. But I don't want to discourage people from just reporting it, because it gives us the opportunity to reach out to that person and say, okay, can you explain what you were seeing, where you were actually sampling, so I please take those sort of macroscopic habitat slime pictures and put those up. But if you have any chance of being able to put these under a microscope, that's the proof is in the pudding right there, or in the slime.
Yeah, if I could just, I don't know, throw out there, it's going to be impossible to make a definitive identification, but if the person can obviously do what they can to reduce glare, possibly get an underwater photo, but something that documents the feathery flocculent nature of the growth, that's one thing that really sets it apart from the native communities, which are kind of just tight and compact to the rocks. They're all still brown and slimy and slick, but the Didymo really have these kind of feathery cotton candy growths, even when they're not clogging the entire stream. There's a photo we showed of the caribou where there were just these tiny little tufts, and they were a couple centimeters longer than what the normal growth is. But that would give us at least better confidence and like, oh yeah, that probably is Didymo versus what maybe the native community is, where it's more like short mowed grass and well behaved.
Thank you. Liz from Lake County is asking if we're willing to do radio interviews. Yes, Mark is willing to do radio interviews.
So is Heidi.
No, absolutely,
We all
Liz. Yes, we would love the opportunity. Doug is remembering that in some of the South Dakota stream work, that they were concerned about trout respiration, specifically with sloughing in stream, the Didymo sloughing in stream and potentially clogging gills. Will this study examine that?
Ooh, that's a new impact for me. I hadn't heard that. I've heard of other diatoms and marine systems doing some of that kind of problematic stuff. Yeah, I think when we get in our streams and at our fisheries stuff, I think taking a peek at a gill flap may be a good idea here.
Yep.
I don't think we haven't got that specifically planned into these projects yet, but definitely concerned about the health of the fishes there.
I think Don had his hand up before Tyler got his question in. So Don,
Okay, I'll be quick so Tyler can ask his question. Mike and Heidi both, I found it interesting that in your first year of the survey that most of your Didymo was found from the caribou on up. I almost would have expected the opposite. Are you looking also for rather than just an assessment, looking for the conditions that influence the growth and why it is where it is?
Yes, we are. And Don, I noticed exactly the same thing when I saw the maps, and I thought, oh my, what is this? Is a northern thing, northern stream thing. We had hoped to get our funding. We had hoped to get all our funding in place to get out there in July last year. We weren't able to get out until the tail end of August, so we again missed a chunk of the growing season. Of course, the growing season is wet conditions, allowed these things to start growing. We got the two sides of the coin here for 2022. Do we want to see it would be if we have a low water year or conditions that are conducive to it, we are going to be out there sampling from. We're going out on whatever May 10th. We're hitting the streams if we can by then, and so I think we're going to be monitoring these at a proper interval to begin to identify what those conditions and changes are that would be conducive to getting this thing in there. And I think
Okay.
last year we just couldn't get going in early enough in the season, and again, it was a surprise to find that often the Didymo seemed to be sloughing off, that it had already had its happy time.
Yeah, I had another quick question. I saw in the literature and some of the press that you talk about how to prevent transferring Didymo from one stream to another. Some of the stuff I saw was like dry it for 48 hours, that sort of thing. But you guys are going from stream to stream during the day. How do you either have 25 pairs of boots, or do you actually have a good way to treat your footwear so you're not tracking it from stream to stream?
Yeah, a great question, Don, and something that we really take seriously in this approach. For us, everything that goes in streams and is headed to another stream, we are cleaning with Virkon, which is a aquatic sterilizing compound, same thing that's used in a lot of the fish hatcheries and things like that. So we are getting our boots in there, all of our sampling gear, to make sure that that we aren't the vector of this. And it brings up a great part of this messaging in this thing is this, how can we convey to stakeholders and people that are visiting the North Shore and using the North Shore that it's something to take seriously. Is it we know that things like felt-soled boots are really a no-no anymore because of their ability to grab microscopic organisms and just move them between habitats. But so it's how do we get that messaging on the street of drain, dry, dispose, and convert it to how we're going to prevent things like this from moving in our own streams. Thanks for that.
Mike. Thanks, Mike and Heidi. Look forward to more information as you collect more, I should say.
thank you, Don. Tyler's question is pretty technical here. What kind of microscope would you recommend for positive Didymo ID?
Really cool ones. David's got one right there. Turn your camera, David, and show your mic. No,
What magnification do you need?
with Didymo, honestly, 100x magnification will give you a clear. Didymo is it's such a big, robust diatom with it's got that sort of that real nice shape to it, and so it jumps out at you when it's there. So I think if you can get just even a dry lens that's giving you 100x magnification through your eyepieces, so just a 10x lens on a microscope should give you confirmation of whether it's Didymo or not. And again, super easy now with cell phone cameras. Just hold your cell phone up there. You gotta get the distance just right and pop the button, and you'll be able to capture enough information that we can see that. But you don't need a super good scope to do it for Didymo.
Even the DNR did it.
One of the real giveaways is the shape. And if you just even look at the button that Mark has on the slide there, I mean, you're just looking at that outline that will give away the identity. There's not a tremendous amount of diatoms that have that shape.
Thank you. Doug provided some additional information about the Didymo clogging gills. It was especially a concern for young of year and larval fish.
Ah, okay.
Elena is asking if we are reaching out to fish license sellers and bait dealers.
We haven't taken that step yet in this process. And again, I think we're at this stage of what messaging do we need to get out there.
And then Joe put a link for just a small pocket microscope in the chat. If you want to check out an inexpensive alternative to magnification. Are there other questions out there, Don? I'm assuming your hand is just still up from before.
Yes, it is. Sorry about that.
That's okay.
Well, I want to encourage people to reach out to us. All our emails are in that initial invitation we sent out, so it shouldn't be too tough to find our names and numbers on there. But please, if you have additional ideas of how we can work together to get out there and access old collections, or get the messaging on the street, or enhance your outreach and education needs, please, that's what we want to do is work together as a team to address this issue. I'm thinking of working on a new button down there with the Didymo on it, and it's going to say, Didymo, it's snot funny. So that's what we're thinking. No one, do I get a laugh of anyone? There you go. Well, as we wrap up the hour here, I do want to make sure I thank everyone, especially our funding agencies. The DNR dive team was great. Rich and Tim, amazing underwater work you're doing. At the station, we've had people already move on. Jeremy was a great help last fall. He's moved on to grad school, but our lab staff there is a big help. Chris at French River made these amazing lobe samplers for us. Nick Peterson was the eyes on the streams for us. Found Didymo where we hadn't seen it, actually, because he had experience with Didymo as well. A few places have allowed us access to the lake shores. Fenstead's Resort, Sugarloaf has, and I forgot to put Lutsen Resort in there, both at the golf course and down at the lodge itself. Brandon up at Grand Portage helped us sample the Grand Portage Creek. State park managers supported our efforts to get permits to work in the state park streams. We had great press from Greg, John, and Joe, and especially just want to thank all the partners out there that are helping us helping us work on this project and get the news on the street. With that, we just surpassed the top of the hour. We reached the top of the hour, and I want to thank everyone. And again, reach out if you have any additional comments, questions, input, or concerns. We'd love to hear from you.